Urinary system. In this video we are going to see how urine is formed. Three processes happen in the nephron to make urine. The first one is the filtration. Filtration occurs in the renal corpuscle, and the blood it filters across the glomerulus, producing filtrate within the Bowman's capsule. So this is the renal corpuscle. We know the renal corpuscle, it consists of the glomerular tuft of capillaries with the surrounding capsule. So because of the hydrostatic pressure inside the capillaries, the fluid, it moves from the capillaries into the space of the Bowman capsule. From there it goes into the tubular system of the kidney and in the of the nephron, and in the tubes two processes are happening. One is the absorption. So tubular reabsorption, it takes place in the tubules of the nephron and the collecting duct, and it involves selective movement of water and solutes from the filtrate back to the blood. So this is a capillary running alongside this tubule, so the substances are being absorbed back into the blood. Then the third process is the secretion. So as tubular secretion, it also takes place in the tubules of the kidney and in the collecting ducts. The substances which are not needed by the body, they are secreted into the tubular fluid. Now let's go over this diagram quickly. This is afferent arteriole. Afferent arteriole, it is coming from the cortical radiate artery. And efferent arteriole, please notice that it is wider compared to the efferent arteriole. So this glomerular capillary bed, it is a capillary bed between two arterioles, and it is a high pressure capillary bed. And now this efferent arteriole, it continues either as peritubular capillary network or as vasa recta, and these run alongside the tubules. The composition of the filtrate and the urine, it is very different, because filtrate, it is almost everything other than the proteins in the plasma, whereas the urine, it only contains the unneeded substances and the excess salt and the metabolic waste. How that is done, that the filtrate, because of absorption and secretion, the composition of the filtrate is changed. Now let's go over these processes in detail. So the first step, it is the formation of the filtrate. First step is the glomerular filtration. Glomerular filtration, it is a passive process. It means it does not require any energy. And then what is the force which drives the movement of the fluid? It is the hydrostatic pressure. It forces the fluid and solutes through the filtration membrane into the glomerular capsule. No reabsorption into the capillaries of the glomerulus occur. So it is only one way. The fluid is going from the capillaries into the capsular space. It is not moving back into the capillaries. No reabsorption is happening here. Now let's go over the filtration membrane. So our filtration membrane, it consists of three layers. First one is the fenestrated epithelium of the capillaries. These are the glomerular capillaries, and this is their fenestrated epithelium. Fenestration means that it has pores. And then it is resting on a basement membrane. This basement membrane is made of the fused basal lamina of the basement membrane of these endothelial cells and of the visceral layer of the capsule. And so after this is the basement membrane, and on the other side of the basement membrane, in the tubular side, we know there are specialized cells which are known as podocytes. These are the food processes of those podocytes, and there are slits between the food processes of those podocytes which are known as filtration slits. These filtration slits, they are also covered by a thin diaphragm, which you can see here, blue line. This is known as slit diaphragm. So it further causes the selection of only smaller molecules are allowed to come inside the lumen of the capsule. So this is our filtration membrane, and because of the pores, it is really porous, but because of the checkpoints, this slit diaphragm and these only small molecules, they can pass through. Also this filtration membrane, it is lined by negative glycoprotein which has a negative charge. So it also stops the negatively charged particles from passing. And the macromolecules that get hung up in the filtration membrane, they are engulfed by the specialized pericyte cells called glomerular mesangial cells. We already talked about them when we were discussing the anatomy. Now let's look at this diagram. This is the afferent arteriole with the wider diameter, and these are the glomerular capillaries, and this is the efferent arteriole. It is smaller diameter compared to the afferent arteriole. So these capillaries, they are lined by the visceral layer of the capsule. The visceral layer of the capsule has specialized cells which are known as podocytes. Those podocytes have cytoplasmic extensions with these foot processes. These foot processes they intervene into each other. There are slit-like spaces between these foot processes which are known as filtration slits. So this is our filtration membrane. And remember those filtration slits, they are also lined by the slit diaphragm, a very thin membrane which also serves as a barrier to big molecules. And then this filtration membrane, it is lined by glycoprotein. That glycoprotein, it stops the movement of negatively charged particles. And this is the parietal layer of the capsule, and we know it does not participate in the filtration. And just as a reminder, this efferent arteriole, it will continue as the capillary peritubular capillary network or as the vasa recta. This is again the same, it is enlarged. This is the capillary epithelium. We can see it is fenestrated, and they have removed the basement membrane, so we can see the fenestrations in the capillary epithelium. And then these are the podocytes. These are their cytoplasmic extensions, and these are their foot processes of the podocytes. There are slits between the foot processes of the podocytes. These fenestrations and these slits, these all, they make the filtration membrane very permeable and porous. Now this is a podocyte, and these are the foot processes of the podocytes. This is the body of the podocyte, and these are cytoplasmic extensions, and these are the foot processes. What are the pressures that affect the filtration? So in the glomerular capillaries, the same pressures that act in the capillaries all over the body, they are the same pressures which cause the fluid to move out of the vessel. So inside the vessel we have hydrostatic pressure. Hydrostatic pressure, it is due to the blood flow, or you can say it is the blood pressure, and this pressure tries to push the fluid out of the vessel into the interstitial fluid. So this fluid is moving into the interstitial space out of the vessel because of hydrostatic pressure. And then there is another pressure which is known as osmotic pressure. Osmotic pressure is due to the presence of proteins and solute inside the capillaries. These try to keep the water inside the vessels. It tries to attract the water because these big molecules cannot leave the vessel. They try to cause osmosis of water inside the vessel. This is known as osmotic pressure. So these are the same pressure which also act on the glomerular capillaries. Now let's talk specifically about the glomerulus, how this glomerular filtration happens. So there are two types of pressures acting on the glomerular capillaries. There are outward pressures which are trying to push the fluid and the solute outwards, and then there is inwards pressure which are trying to push the fluid and solute into the capillaries. So let's see what are the outward pressure. So the outward pressure is the hydrostatic pressure in the glomerular capillary. This GC, it represents the glomerular capillary, and it is basically the blood pressure. The more the blood pressure there is, the more it will be pushed. It will try to push the fluid and solutes into the capsular space. It is around 55 millimeters of Hg. It is in the glomerular pressure, this capillary bed, it is high pressure bed. Why? Because it is being drained by the efferent arteriole. Efferent arteriole is a high resistance vessel with a diameter smaller than the afferent arteriole. So it makes it more a high resistance. That's why the pressure in this capillary bed is high, and filtration, it occurs throughout the length of the capillaries. The inward pressures, which are trying to push the fluid and solute into the capillaries, they are the hydrostatic pressure in the capsular space. The CS, it is for the capsular space. It is 15 millimeters of mercury. Then there is colloid osmotic pressure in the capillaries, which is 30 millimeters of mercury. So the proteins and the solutes in the capillaries, they try to pull the water back in. The osmotic pressure, they try to cause the osmosis. And we know, like, there are no proteins in the filtrate, so there is no that solute pressure or osmotic pressure present over there. And how do we calculate? We calculate it by the net filtration pressure. Net filtration pressure, it is a sum of all the forces. So 55 millimeters of mercury, which is forcing out, minus the 45 millimeters of mercury which is getting in. So 30 plus 15 is equal to 45. And when we subtract 45 from the 55, we get 10 millimeters of mercury. And this is the pressure which is responsible, 10 millimeters of mercury, which is responsible for the filtrate formation. Glomerular filtration rate, it is the volume of filtrate formed per minute by both kidneys. Normally it is 120 to 125 ml per minute. And we use it to determine the functioning of the kidneys, that if the kidneys are functioning good, this is how we see if the glomerular filtration rate is good. Glomerular filtration rate is directly proportional to net filtration pressure, total surface area available for filtration, and the filtration membrane permeability. So if any of these increase or decrease, the GFR will increase and decrease. The net filtration pressure, it is dependent upon the glomerular hydrostatic pressure, and this is a controllable factor. How can we control it? How can we control the glomerular hydrostatic pressure? By changing the diameter of the afferent arteriole, and sometimes also of the efferent arteriole. Remember, the arterioles, even in the systemic circulation, their diameter can be changed. If they constrict, they become smaller, and if they dilate, they become bigger. So this is a controllable factor, the diameter of the arteriole. Then the total surface area available for filtration, it can be controlled by the mesangial cells. The mesangial cells which we saw, the specialized cells, they can increase or decrease the surface area available for filtration by contracting. And then the filtration of the membrane permeability. We know the membrane, it is permeable due to fenestrations. This permeability will increase or decrease in diseases. It is not a factor which we can increase or decrease. Now let's go over the regulation of glomerular filtration, how our body, it regulates the glomerular filtration, how it keeps it within a certain range. And why does the body need to regulate the GFR? It is because the kidneys, they need to do it so that they can maintain extracellular homeostasis, so they can have normal filtrate. And the body, it regulates the GFR to manage the blood pressure. And this is done by two ways. The glomerular filtration rate, it is managed by two ways. One is the local controls, and local controls, these are the renal autoregulation, and this is what is used by the kidney. And the other method, they are the extrinsic controls. Extrinsic controls, they are used to manage the blood pressure. Now let's go over the intrinsic controls. Intrinsic means inside. So they are the controls which are in the kidneys. So kidney is auto-regulating. That is why we call them renal auto-regulation. So intrinsic controls, they maintain nearly constant GFR when mean arterial pressure is in range of 80 to 180 millimeters of Hg. This is when these controls, they operate, and they maintain the glomerular filtration rate. And auto-regulation, it ceases if it is out of that range, because then the body, it wants to stabilize the blood pressure. So then and the extrinsic controls, they overcome the intrinsic control. Intrinsic controls don't work if the blood pressure is not in this range, because then the primary goal of the body becomes to maintain the blood pressure before the glomerular flow is maintained. So the two types of renal autoregulation mechanisms are, there is myogenic mechanism and then there is tubuloglomerular feedback mechanism. Myogenic mechanism, it uses the property of the smooth muscle. Then when it is stretched, it contracts. So when there is high blood pressure, the afferent arteriole, it is stretched, and we know there is smooth muscle in the afferent arteriole. When afferent arterioles, they are stretched, they constrict. In this way, they restrict the blood flow into the glomerulus, and they prevent the glomerulus from damage due to high blood pressure. Similarly, the decreased blood pressure, it causes dilatation of the afferent arterioles, and they both help to maintain the net filtration pressure and the glomerular filtration rate despite normal fluctuations in the blood pressure. The other mechanism of the autoregulation is known as tubuloglomerular feedback. Let's go over this. This is our renal corpuscle. Renal corpuscle consists of glomerulus and the Bowman's capsule. This is the capsular space. This is where the filtrate is being made. So just imagine when there are lots of glomerular filtrate is being made, it means GFR is high, and because of this GFR, the filtrate, it moves with a kind of rapid space through these tubules. And when the filtrate is moving through the tubules, we know that certain electrolytes and water are reabsorbed, and certain things are secreted before it is removed from the body in the form of urine. So sodium, it is absorbed in this, especially in this segment. So when the filtrate, it is passing through this portion of the body, the sodium, it is reabsorbed into the blood. And if the GFR is high and the filtrate is flowing with a more than normal speed, or due to some other reason, this sodium is not reabsorbed, when that filtrate reaches this point, there is increased sodium in the filtrate, and which is detected by the macula densa. What is macula densa? Macula densa, it is present in the thick ascending limb, especially with the thick ascending limb. It converts into the distal convoluted tubule, and it is in closed proximity, in juxta to the afferent arteriole. So these specialized cells, they are chemoreceptors. They can detect that there is too much sodium in the filtrate. What do? They cause constriction of the afferent arteriole. When the afferent arteriole, it constricts, the blood flow in the glomerular capillary decreases. As a result, the NFP decreases and GFR decrease. Now it allows more time for the sodium to be reabsorbed, because the speed has decreased. And it occurs the opposite when GFR is too low. Now let's go over the extrinsic controls. So remember, the extrinsic controls, their primary purpose is to maintain the blood pressure. Their primary purpose is not to maintain the GFR. They are maintaining the GFR to maintain the blood pressure. So the extrinsic controls are neural and the hormonal mechanism. Purpose of extrinsic control is to regulate GFR to maintain systolic blood pressure. Extrinsic controls will override the renal intrinsic controls if blood volume needs to be increased. Okay, so these are two. They are sympathetic nervous system And then we have renin, angiotensin, aldosterone system. So this is our nervous neural control, and this is renin, angiotensin is our hormonal control. Let's go over these. So sympathetic nervous system, under normal resting conditions and under normal, when everything is going normal, the renal blood vessels, they are dilated, and renal autoregulation mechanism, they prevail. So under normal blood pressure conditions, normal conditions, the renal autoregulation mechanisms, they are in charge. But what happens when things go wrong? Under abnormal conditions such as extremely low ECF, when the volume is very low, the blood pressure is very low. Now the body needs to shunt the blood to go to the vital organs. So now what happens, that norepinephrine, it is released by the sympathetic nervous system, and epinephrine is released by the adrenal medulla due to baroreceptor reflex. Remember, when the blood pressure is low, the baroreceptors, they can detect, and when the blood pressure is high, the baroreceptors, they can detect, and then the signals are sent to the appropriate systems in the body. So now we have secretion of the norepinephrine and the epinephrine. And what do? The sympathetics, they always vasoconstrict. When they vasoconstrict, they increase the blood pressure. So there is systematic vasoconstriction, and then there is also constriction of afferent arterioles in the kidneys. And when afferent arterioles, they are constricted, the GFR is decreased, and the blood volume and pressure, they increase. Sometimes this constriction can be so much that the kidneys can get damaged, their blood supply can be compromised. But at this time, because the blood pressure is very low, the body wants to shunt the blood to the vital organs, to the brain, to the heart. So this is so it constricts the afferent arteriole in other parts of the body, including in the kidney. In the kidneys also, the afferent arterioles, they are constricted. Now the other mechanism is the renin, angiotensin, aldosterone mechanism, and we are all familiar with this because we have been going over it since the beginning of this course, from endocrine, then in the cardiovascular system, and now this is the last part of it. So renin, angiotensin, aldosterone mechanism, what does it do? It maintains it. It is the main mechanism for increasing blood pressure. And how does it do? How does it do it? It releases renin. Renin is released by 3 pathways. Okay. So what is the first pathway? First pathway is the direct stimulation of the granular cells. First of all, the granular cells are going to release the renin. These are the granular cells, right? And they are the cells which are of the afferent arteriole, and they contain granules. Those granules contain renin. All right. So these are the cells which release the renin. There is direct stimulation of the granular cells of the juxtaglomerular complex. This whole thing is known as juxtaglomerular complex by the sympathetic nervous system. So the sympathetic nervous system, it stimulates these cells, and they secrete the renin. And then, so these are the granular cells and sympathetic nervous system. They stimulate to secrete the renin. And then the other process is, when there is low blood pressure, there is reduced stretch. There is reduced stretch of the afferent arteriole, and these cells, they detect it, and hence they secrete the renin. They know it is time to secrete renin. And then what happens? What is the other mechanism? That there is stimulation of macula densa, which when filtrate NaCl concentration is low. Why is NaCl low? NaCl is low because blood is flowing very slowly in the tubules, and too much sodium is being reabsorbed. So this also tells the macula, that the sodium is low, and then it stimulates the granular cells to secrete the renin. So these are the 3 processes which end up in the secretion of renin. And that renin, we know then it converts angiotensinogen into angiotensin 2, and which causes the release of aldosterone. Now there are also other factors that renal cells, they release a variety of chemicals. Some chemicals act as paracrines that affect the renal arterioles, such as adenosine and prostaglandin E2, and some cells make their own locally acting angiotensin 2, and this reinforces the effects of hormonal angiotensin 2. Angiotensin 2 is a vasoconstrictor itself, and it also causes the release of aldosterone. Now let's go over this chart so we can sum up everything. When the systemic blood pressure is low, let's first look at these intrinsic mechanisms. These are the intrinsic mechanisms, this blue and the peach column in the start. So when the systemic blood pressure is low, blood pressure in the afferent arteriole is low. As a result, GFR is low. So there is decreased stretch of smooth muscle in the wall of the afferent arterioles. This leads to the vasodilation of the afferent arteriole. And when afferent arterioles are vasodilated, more blood goes into the glomerular capillaries, which increases the GFR. Now let's see in this column. So when the systemic blood pressure is low, GFR is low. That GFR leads to decreased filtrate flow, and when the speed of the filtrate flow is decreased, so more of the sodium chloride, it is reabsorbed, and the sodium chloride in the ascending limb of nephron loop is decreased, and this acts on the macula densa cells in the juxtaglomerular complex of the kidney, and those macula densa cells, they cause the release of they inhibit the release of vasoactive chemicals, and they cause the vasodilation of the afferent arterioles, because those macula densa cells, they know that we need to increase the blood flow, so sodium levels are not so low. This also increases the GFR. Now let's see what's happening in the extrinsic control. When the blood pressure is low, the granular cells of the juxtaglomerular complex of the kidney, they release renin. And we know that what are the three mechanisms which cause the release of renin. That renin is released by these cells, and this renin, it leads to formation of angiotensin. So angiotensin 2 is made from angiotensinogen, and there are many steps in this process, but the end product is angiotensin 2. We got angiotensin 2. Now this angiotensin 2, it is a direct vasoconstrictor of the systemic arterioles, and when it vasoconstricts the systemic arterioles, the peripheral resistance increases, which leads to increase in blood pressure. Then we know it also acts via aldosterone, which is secreted by the adrenal cortex, and that aldosterone, it causes the increased absorption of sodium from the kidney tubules, and water loves sodium. So when sodium is reabsorbed, water follows it, and this increases the blood volume. This also leads to increased systemic blood pressure. And then let's see how the neural control is happening. So the systemic blood pressure, it inhibits the there is low blood pressure, so the baroreceptors are inhibited in the blood vessels of the systemic circulation. This leads to sympathetic nervous system. We know that they are balanced, the parasympathetic and the sympathetic, they balance each other. So there is sympathetic nervous system stimulation, which leads to vasoconstriction, and then this way systemic blood pressure, it is restored. This table summarizes everything we went over, so you can go over it. Now, what can go wrong in GFR, in the glomerular filtrate? So anuria, it is a term when the urinary output is very low, when it is less than 50 ml per day, and it indicates that the glomerular blood pressure is too low to cause filtration. So renal failure and anuria can also result from situations in which nephrons, they stop functioning. For example, in acute nephritis, when there is inflammation or infection, or in transfusion reactions, this can occur, and this can also occur in crush injuries, that the nephrons, they are not working at all and they are not making urine, or they are making very less urine. So the term is anuria.